Purification of genomic DNA from bacteria. So this session is going to look at the basic methodology that has been used for years together to isolate DNA. And it is important to understand how these basic steps lead to finally purification of DNA. Of course, there are a lot of techniques now or methodologies that have been developed today, which is also enabling purification of DNA in just one single step. So let us look at the learning outcomes of this session. So genomic DNA isolation is typically divided into 4 steps, with each step leading to separating DNA from all the other cell components. Sufficient biomass is harvested and processed to get a cell extract, and then centrifugation is carried out to obtain the pellet, uh, or is used basically to ensure that eventually one gets the pure DNA, and therefore centrifugation plays a very important role in the process. Phenol chloroform is used to denature proteins, and the DNA RNA are precipitated using appropriate solvents. Now, applicability of this technique in analyzing nucleic acids is very important. So, let us look at the basic steps that are involved in isolation and purification of DNA. So, what is generally done when one needs to isolate DNA from bacteria is to grow bacteria. And once one reaches a certain biomass, then that bacteria is centrifuged or the broth is centrifuged to harvest the cells. Once the cells are obtained, these cells are then resuspended in an appropriate buffer, and having resuspended it, these cells are lysed to get the cell extract. So that becomes the second step. Once the cell extract is obtained, from this cell extract, only the DNA is to be purified. So there are different ways of ensuring that either the other components are denatured and removed, or the other components are separated from the DNA. So one gets pure DNA, and this pure DNA can be concentrated further by certain methods. So the 4 steps basically is growing the bacteria and harvesting it, removing or breaking the cell wall and the cell membrane to get a cell extract, purifying the DNA from the extract, and then further concentrating it. These are the 4 basic steps. Now what we are going to look at is look at each individual step and see the um, necessary molecules or chemicals that are used to carry out this entire process. So the first step is growing and harvesting of bacteria. Now, generally, whenever one needs to grow or harvest bacteria, a suitable nutrient media has to be selected. So the bacterial culture is established by growing the bacteria either in defined media. So defined media is when you know each and every constituent that is being added to the media, then that is what is called as defined media. And one of the examples of defined media is an M9 media. One can also grow the bacteria in undefined media. So for example, Luria bertani or the nutrient broth, they are undefined media which contain certain constituents such as tryptone and yeast extract. And in these tryptone and yeast extracts are basically protein digests. So there are innumerable amino acids present, but one is not sure how much or what is the defined amount of amino acids that are present, and hence the media becomes undefined. Now, if one just has to get DNA from the bacterial source, then undefined media is enough, or is what is needed. But if, so say for example, a specific study is being carried out where one needs to know exactly what is there in the media, then only for such specific studies the defined media are used. So the growth of culture is of course monitored by measuring the OD at 600 nanometer, as the culture becomes more and more turbid, the OD of the culture is going to increase. Say, for example, you are looking at E. coli, then with E. coli, what has been observed is because it multiplies every 20-25 minutes, the OD of about 1, when the culture reaches the OD of 1, it is equivalent to containing zero point 0.8 into 10 raised to 9 cells per ml. And an ideal biomass for DNA isolation is about 2 to 3 into 10 raised to 9 cells per ml. So therefore an OD of 1 or a little more is suitable for DNA isolation from bacteria. That biomass is good enough to give a few nanograms of DNA. Now the cells preferably harvested are during the log phase of the bacterial culture. This is something that most try to take care of, that when the bacteria is still in the log phase, it should be harvested. There are two reasons for that. One, when it is in the log phase, of course, you have the bacterial cell multiplying and therefore you have the DNA doubling. So, therefore, a logarithmic growth phase will have a sufficient amount of biomass. When suppose this culture goes to the stationary phase, in the stationary phase, you will have viable cells as well as non-viable cells. And so in the non-viable cells, there are possibilities of DNA getting fragmented or damaged. And hence, when you isolate the DNA, you will get a sufficient amount of DNA, but then the DNA can be fragmented and not the intact genomic DNA. Therefore, preferably, it is harvested or the cells are harvested when in the log phase itself. Once it is collected in a centrifuge tube and centrifuged, one will definitely get the bacterial pellet and the media constituents with other contaminants may be present in the supernatant. The supernatant is discarded, and after discarding the supernatant, the pellet of bacteria is resuspended in a buffer that is called as tris NaCl EDTA. Tris allows maintaining the pH at 8. EDTA is important because it helps in the next step. We will see how. And NaCl maintains isoosmolarity, so therefore the cells do not lyse at this stage itself. So the bacterial cells are collected as a pellet after centrifugation, with the supernatant being discarded, and as mentioned, the pellet is resuspended in buffer, the buffer generally being TNE, that is tris NaCl EDTA. The next step is preparation of a cell extract. So we all know that bacteria have, apart from the cell membrane, a cell wall surrounding it. Now, in certain cases, one also has the capsule around the cell wall. So therefore, most bacteria will have a cell envelope. If it is gram-positive bacteria, then it will have a thick cell wall made up of peptidoglycan layer. So peptidoglycan layer is generally a polymer made up of a repeating unit of a disaccharide, that is basically, and the disaccharide unit basically comprises N-acetyl glucosamine in a glycosidic linkage with N-acetylmuraminic acid. So this disaccharide keeps repeating to form one peptidoglycan polymer, and then you will have several peptidoglycan cross-linked to each other by peptide linkages. So that is how one gets a thick layer of peptidoglycan which is present in the gram-positive bacteria. In the gram-negative bacteria, although the peptidoglycan layer is thin, other than the peptidoglycan layer, the gram-negative bacteria has an outer membrane of lipopolysaccharides. So be it the gram-positive bacteria or be it the gram-negative bacteria, it is important to disrupt the cell wall and then finally disrupt the cell membrane. So one can disrupt both of these through physical methods. So physical methods would basically involve mechanical aberrations, but that can also lead to damaging the DNA per se, and hence it is a less preferred method. Most methodology involve using chemical methods, and in chemical methods there are two agents that are generally used. One agent that is able to disrupt the cell wall, and the second agent that is able to disrupt the cell membrane. Now, in order to disrupt the cell wall, in case of bacteria, generally what is used is EDTA and lysozyme. So, like we mentioned, the cells were suspended in TNE, that is tris NaCl EDTA. So, the EDTA that is present in the buffer is actually weakening the cell wall. So, EDTA has a dual role to play. One, it is able to chelate the magnesium ions, and on chelating magnesium ions, the peptidoglycan layer or the cell wall of the bacteria becomes weak. Addition of lysozyme. So, when lysozyme is added, lysozyme is an enzyme that is going to break the glycosidic linkage between N-acetylglucosamine and N-acetylmuramic acid, and thereby it is again going to disrupt the cell wall. So combination of EDTA and lysozyme is good enough to disrupt the cell wall. The second agent that is used is the one that is used to disrupt the cell membrane, and most commonly what is used is sodium dodecyl sulfate. This is an anionic detergent and it is a strong detergent. Detergents being amphipathic in nature, they're able to interact with the lipids in the bilipid layer and remove the lipids from the bilipid layer, thereby making the cell membrane leaky and also therefore eventually leading to complete disruption of the cell membrane per se. So you can see how using a chemical method you can first disrupt the cell wall and then disrupt the cell membrane. With both having been disrupted, obviously cell lysis happens, and cell lysis gives rise to formation of what is called as cell extract. Now, in case of bacteria, there being no other membranous structure other than the cell membrane, therefore what is interesting is that all biological components present inside the bacterial cell will be present in the solution. Hence, on centrifugation, what is going to happen is all the cell debris will settle down, and in the supernatant will be the biomolecules, which include the DNA, RNA, and protein majorly in the bacterial system. Now, therefore, what is required to be done is from this supernatant, one has to purify or isolate the DNA. So the step 3 is basically purification of DNA from the cell extract. Now, how can one carry this out? There are two basic strategies. One strategy is to remove all the other components except DNA. So if you have the cell extract will have DNA, RNA and proteins. So in this case, you will consider the RNA and proteins as contaminants. In bacterial cell, you will have carbohydrates but in much lesser amount. So carbohydrates do not become a very major contaminant in case of bacteria, except if one is dealing with bacteria that exudates polysaccharides. So for example, Pseudomonas can give rise to exopolysaccharides, and in such cases, what happens is one gets very slimy cell extract. Then from that getting DNA becomes a little difficult. But otherwise, from most other bacteria, one has very less carbohydrates, and so one can individually now remove the proteins, carbohydrates, and the RNA to now get a solution which only has pure DNA. So this is the strategy where you're eliminating the contaminants one after the other. Most common method to remove these contaminants is by organic solvent extraction. Now, we know that the cell extract is an aqueous solution, and so therefore an organic solvent when used, it brings about what is called as phase separation. So the most widely used organic extraction is using phenol chloroform, with isoamyl alcohol. Now, phenol and chloroform is used generally in the ratio of 1 is to 1. And in the phenol chloroform, the phenol that is used is buffered phenol. So the pH of the phenol is brought to about 8, because what is observed is very acidic phenol can also tend to denature the DNA, and therefore to prevent that from happening, buffered phenol is used. Now, both phenol and chloroform, being organic solvents, they make a phase which will be very separate from the aqueous layer. And that is a very important thing in organic extraction. So, if you have the cell extract now has protein, DNA, RNA. Let's say they are the major constituents, and you may have carbohydrates and lipids. Lipids obviously will get solubilized in chloroform and phenol. So they will go with the organic phase. The DNA, RNA, and the protein are soluble in water, and so they may remain in the aqueous layer unless and until they are denatured. So when phenol chloroform is added to the cell extract and mixed well, then the proteins that get exposed to the phenol get denatured. That is because phenol is able to affect the protein folding, and by affecting the protein folding, the 3D structure of the protein is disrupted, and thereby the proteins begin to aggregate. So you would basically have proteins getting coagulated. Now, when this mix with the phenol chloroform is centrifuged, on centrifugation, the denser solvent is going to be the phenol, and therefore you will have a layer of phenol chloroform over here. You will have one layer of the at the interface of the aqueous layer and the organic layer, and that interface is that of the coagulated proteins, and you will have the aqueous layer at the top, which contains the DNA and the RNA. So therefore, after the phenol chloroform step, you will on centrifugation get 3 layers. I'm repeating. You'll have the aqueous layer at the top containing the DNA, RNA. At the interface of the aqueous and the organic layer, you will have organic solvent layer, you will have the coagulated proteins forming a buffy layer, and then of course you will have the phenol chloroform organic solvent layer. This will contain lipids, carbohydrates, and small polypeptides. Now, interesting to notice, when chloroform is added at this point, the chloroform is accentuating the phase separation. So therefore, the addition of chloroform with phenol is ensuring that you have a clear, um, phase separation happening. Because the phenol here that is used is buffered phenol, and because it is buffered phenol, the phenol can tend to be slightly miscible with the aqueous layer. So in order to ensure that you have proper phase separation, chloroform is definitely added. Now, once you get this, what is important is to collect the aqueous layer. Now, once the aqueous layer has been collected, many a times, many protocols have the aqueous layer again being treated with chloroform, mixed well, and centrifuged again to get a layer of aqueous and a layer of chloroform at the bottom. This is just to ensure that if there is any phenol present in the aqueous layer, it gets into the chloroform layer. And secondly, if there is any protein, chloroform can again denature the protein, and you can get a buffy layer again at the interface. Now, if the contamination of protein is found to be greater, then the phenol chloroform step can be carried out more than once. Please to understand that with more number of phenol chloroform steps getting repeated, you would definitely have a purer sample of DNA obtained, but you may also have lesser quantity of DNA obtained in the final sample. In many cases or in many protocols, what has been obtained or observed is before carrying out the phenol chloroform step, the cell extract is pre-treated with proteases such as proteinase K, And when that is done, then the proteins are broken down into smaller polypeptides, and these smaller polypeptides are much easily extracted by the phenol. So, to ensure a greater removal of the proteins, this proteinase K treatment is carried out. Most mRNA will of course be present with the DNA, and so if in your particular study you do not want any RNA, then you have to treat the aqueous layer that is obtained here with RNases to remove the RNA. Thereafter then this aqueous layer will only contain the DNA. Now the next strategy, or another strategy that can be used for purification of DNA from cell extract, is to separate the DNA from the other components. So the previous strategy was to eliminate the other components from DNA such that only the DNA remains in solution. And in this case, what is done is that from the extract, the DNA is separated from all the other components. So, the most preferred method has been ion exchange chromatography. The reason for using ion exchange chromatography is that in bacterial systems, if you look at whether you take proteins or DNA or RNA, all of them are negatively charged and they will have a tendency to bind to a ion exchanger. So, the basic understanding is that the resins, which are ion exchangers, will have a higher affinity for DNA or RNA or proteins, and the affinity of these would vary depending on the charge that is present in them. If one adds salt, then this affinity with which the DNA, RNA, or protein binds to the ion exchanger gets affected, leading to release of the biomolecules association with the resin. So elution can be carried out by adding increasing salt concentration. So therefore, in column chromatography, what is therefore done is an ion exchange resin is taken and the cell extract is loaded. Once the cell extract is loaded and allowed to run, the DNA, RNA and proteins all will get bound to the ion exchange resin. Now what is done is different concentrations of salt solutions are added. So at a low salt solution, you will have the protein and the RNA eluted out, while when you add a greater amount of salt, then only the DNA will be eluted out because the DNA binds to the resin with a greater affinity than the RNA and the protein. So sequentially, one can remove the proteins, the RNA, and then one can remove the DNA from the resin to get pure DNA collected in the fraction. So as mentioned, all negatively charged molecules bind to the resin with different affinities, and that is what is taken as a strategy to purify the DNA. The last step, of course, is concentrating the DNA samples. Now, the most common methodology is ethanol precipitation, or one can use any other organic solvent as well, laced with a little amount of salt. So, chilled ethanol is generally used with monovalent salt ions. So, what happens is with monovalent salt ions, the precipitation is enhanced. Now, how does the chilled ethanol and the monovalent salt ions help in precipitating out the DNA? We all know that DNA is soluble in the aqueous layer because the DNA forms a hydration layer around it. Now, when one uses chilled ethanol and monovalent salt ions, then that hydration layer around the DNA is disrupted. The DNA gets more and more dehydrated, and therefore that will get precipitated out. Short chains and monomeric nucleic acids do not precipitate that easily, but if you have a longer DNA, it will definitely precipitate out very fast. Two ways of ethanol precipitation is possible. One is that you have the cell extract with the DNA and you layer it with the chilled ethanol. Now, at the interface, what one will slowly see is that the DNA begins to start getting converted into long filaments or long thread-like structures. So when you add a glass rod inside and swirl it, okay, you can spool out the DNA. So the DNA will get attached to the surface of the glass rod, and as you take out the glass rod, you will only find the DNA attached to the glass rod. So that then you can resuspend that DNA in an appropriate buffer. The other way is, of course, once you add the ethanol, you mix it well with the extract and then centrifuge it. So, when you centrifuge it, the precipitated DNA will get collected as a pellet and the ethanol will remain as a supernatant. This ethanol is discarded, the pellet is allowed to air dry, and then this pellet is resuspended in either distilled water or intrus EDTA buffer. Now, the addition of EDTA in most of the buffers that is involved in isolating DNA is, for one, initially the reason was that it is weakening the outer envelope of the bacteria. But the other reason for using EDTA is that EDTA being able to chelate divalent ions, especially magnesium ions, uh, it is it is preventing the DNA from getting degraded because nucleases generally are able to degrade DNA, but nucleases are active only when they have or they are bound to the cofactor magnesium. So with EDTA being present, magnesium is chelated to EDTA, and so the nucleases become inactive, and so the DNA integrity is maintained. Therefore, EDTA is commonly used in all the buffers in isolation of DNA. However, at this point, it is important to note that if one uses this DNA for further studies wherein a different buffer system is needed, then the tris-EDTA may interfere. And so in such cases, the DNA is uh, suspended or redissolved in distilled water itself. Now, an alternate method, there are of course several, but one of the most common alternate method that is used is guanidinium thiocyanate. Now, using of guanidinium thiocyanate denatures and dissolves all biomolecules except nucleic acids. Look at how it works. It is denaturing and dissolving all other biomolecules except nucleic acids. And this is pertaining to the first strategy that we looked at for DNA purification. The second important facet about guanidinium thiocyanate is that the DNA, when it is present with guanidinium thiocyanate, the DNA has a higher affinity to bind to silica. And so, uh, silica can be a means of allowing the DNA to attach to the silica. So, the silica can be collected and everything else can be removed. So, all the other contaminants, that is proteins, RNA, will remain in solution, and what will remain bound to on the silica is going to be the DNA. So therefore, what is generally, and of course, when you want the DNA to be separated from the silica, you can always use salt. So the salt will detach the DNA from the silica and you will get the DNA from the silica. So what is generally done is that the silica is used to form a column, okay, the matrix of the column, and the cell extract is loaded on this. So when the extract goes through the column, of course, one must remember that the silica is either made in guanidinium thiocyanate or the cell extract contains guanidinium thiocyanate. So we see that in the presence of guanidinium thiocyanate, the DNA will have a higher affinity to bind to the silica. So when one adds water, all the other denatured biomolecules, that is protein, RNA, etc., is going to flow out. Okay, that is the first thing that is going to flow out. Even without adding of water, when the cell extract is added, you have this getting added. Now, what remains in the silica is only the DNA. So when one adds water or a side or a dilute salt solution, then this time what is eluted out is the pure DNA. And this pure DNA then can be concentrated using ethanol precipitation. So these are the general methods that is used for isolating the DNA. Now, having obtained the DNA, what is important to find out is how much amount of DNA has been obtained. So measuring the amount of DNA isolated is very important. Why is it important? First, you definitely get the yield. So you know how effective your isolation protocol is. Secondly, if you want to use the DNA for further studies such as cleavage by restriction endonucleases and gene cloning, etc., then it is imperative to know how much amount of DNA is present, because the enzyme that is going to be used will have to have a certain amount of activity to cleave or cut the DNA. So, therefore, the amount of DNA will say how much amount of enzyme one has to use. So, that becomes one instance, and likewise, knowing the amount of DNA can help in various further studies. But what is also important is that along with knowing the amount, that is quantity of the DNA, one also has to take care of assessing the quality of the DNA. Now, when we talk about quality of the DNA, what do we mean? We mean whether the sample of DNA that you have got is pure DNA or it has impurity in it. Now, how does one measure the amount of DNA isolated? So, the most common method that is used is, of course, using UV visible spectrophotometer. The reason for using UV spectrophotometer is because DNA absorbs light in the UV range. So, as we can see over here in this graph, all the four nitrogen bases, whether it is adenine, guanine, thymine, or cytosine, they all absorb light around 260 nanometers. So, on an average, a DNA which has all the four nucleotides will absorb light maximally at 260 nanometers. So, therefore, this is the absorption maxima for DNA. And it has been noted that for a 1 centimeter path length in the UV spectrophotometer, an optical density at 260 nanometer, when it equals to 1, then that is for the following solution. So a 50 microgram per ml solution of double-stranded DNA will have OD of 1, or a 33 microgram per ml solution of single-stranded DNA will have an OD of 1, or a 20 to 30 microgram per ml solution of oligonucleotides will have a OD of 1, and a 40 microgram per ml solution of RNA will also have an OD of 1. So if you know that your sample contains an OD of 1, then that sample will contain 50 microgram per ml of double-stranded DNA. So you can actually calculate the amount of DNA present in your sample by just finding out the OD at 260 nanometer. And then you would find the quantity by just comparing it with a standard. What is the standard? 1 OD equivalent to 50 microgram per ml solution of double-stranded DNA. If you are studying RNA, then 1 OD is equivalent to 40 microgram per ml solution of RNA. So if your sample is 0.05 OD, what will be the amount of RNA present in that sample? And like we said, that it is also important to find out the purity of the nucleic acid sample. So that is carried out by finding out the ratio of the sample at OD260 by OD280. Why is 280 taken? Because at 280 nanometer, proteins absorb. So the maximum absorption maxima of proteins is 280 nanometers, and the major contaminant could be either protein or it could be RNA. But RNA also absorbs at 260 nanometer. However, if OT of 260 by 280 is 1.8, then it is indicative of being a pure DNA sample. But if the OD 260 by 280 is about 2, then it is indicative of having pure RNA sample. If the OD 260 by 280 ratio is lesser than 1.8, then it indicates that it has protein contamination. So if that sample has a ratio of lesser than 1.8, you cannot use it for, say for example, PCR, or you cannot use it, say for example, for RE digestion. Then one needs to carry out protein chloroform uh, step again to remove the proteins that is present in the sample. That should be continued until you get a ratio of 1.8. So therefore, this is a gold standard of finding out what the quality of DNA is in the sample that you have got. Of course, now we come to this final question, is why isolate the DNA? So time and again I have mentioned that if you want uh, to carry out PCR, or if you want to carry out RE digestion, then you need DNA. But let us look at a larger or a bigger picture of why you need to isolate the DNA. So DNA is needed if you want to carry out genetic engineering. So any gene cloning study, and you need the DNA. Why? Because from the DNA you will get the gene. That gene of interest you will put into a vector, and that vector is going to be multiplied. So that is gene cloning. You will also need it for transgenics, recombinant products, etc. So DNA isolation is important for the fact that any genetic engineering needs to have the DNA at the front. DNA can be used in forensic sciences. This is a very well known thing. One wants to identify a criminal, then the DNA matching is done, and with that one is able to find out whether the sample that is there is matching the sample of the criminals. One can also do paternity testing by the DNA. DNA can be used to identify species, so not just the percentage GC, but also finding out the sequence of the DNA, and the sequence of the DNA in turn can give an identification of the species. So many bacteria and viruses are identified by carrying out the DNA sequencing. Gene therapy, it can definitely be used. It can be used for evolutionary studies, it can be used for sequencing, and we know that when all DNA is sequenced, then you have the study of what is called as genomics. That means a study of all the genes in a cell is what is genomics. It can be used for nanobiology. So one has seen that a lot of DNA origami structures have been created for using in nanotechnology. Genomics has also, of course, led to enabling use of DNA for diagnostics and drug development. So, this is a bigger picture of where DNA is required or used, and therefore it is important to isolate the DNA. So, let us make the conclusions. The genomic DNA isolation involves growth and harvesting of cells, cell lysis to give the cell extract, and then separation of DNA from all the other components. In bacteria, the major components in cell being DNA, RNA, and proteins, the basic protocol includes removal of proteins, which is definitely carried out by the phenol chloroform step. The isolated DNA is precipitated using chilled ethanol. Many cases, isopropyl alcohol is also used, and then assessed for quality and quantity. And the quality and quantity are assessed by UV spectrophotometer by taking the OD at 260 and at 280 the nanometers. The isolated DNA can be used for various applications, especially related to disease, diagnosis and drug development, and of course, formation or production of recombinant products. The basic methodology, therefore, or strategy in DNA isolation is in eliminating other components or separating other components from DNA. There are many rapid methods developed in the past decade for research purposes. Like mentioned earlier, now you can carry out DNA purification even in one step. Thank you.